share. Okay. Can you see my screen? Are we good?
Yes, sir.
Okay, thank you. Thank you, Ma'am AKR, for that introduction. And thank you for inviting me. And thank you to the Microbial Consortium of the Philippines and the PSM for the invitation. And thank you to all of you. I know it's a Saturday morning. I'm sure it's nice there and sunny, and you have better things to do on a Saturday morning. But I really appreciate your time, and I'm excited to talk to you about one of the things that I really enjoy talking about, which is bioreactor experiments. So let's start with a few questions. Anytime I'm faced with a new technique, a new technology, a new research opportunity, the first thing I ask is, should I do it? Should we do this? Right? So it's a question of value. Is this something that's valuable? Is this something that would actually be beneficial to my students, to my research program? That's the first question. Then the second question is, can we do it? Do we have the means? Do we have the capabilities? Is this something that is really possible in our situation? And then the third question then is, how do we do it? So if you say yes to the first one and you say yes to the second one, then you think about, okay, how do we start doing this? So I bring this up because sometimes engineers and scientists start with the first question, can we do it, right? It's a question of, this is something new, exciting, can we do this? And I'd like to flip it around. And the first question to me is this something that we should do? So it's a question of value. And of course, I'm talking about bioreactors. So what I want to do today is convince all of you that if you think this is of value to you, if your answer is yes to the first question, the answer to the second question is also yes, you can do this. And then maybe today we'll talk a little bit about how you can do this. Okay. So bioreactors and microbiology, like academician Raimundo mentioned, I am an engineer, but I'm also a microbiologist. I'm on the faculty of microbiology here also at NC State. And so I work at the interface of engineering and microbiology. And so we'll go through some examples, maybe establishing the value of bioreactors. So the classic example would be wastewater treatment. And the questions that you might want to ask are, how do you improve the microbiological processes for making human waste and industrial wastewater safer? So treated to the extent that it's no longer polluting the environment and safe for humans. And make it more sustainable, which means it's safer, it's more efficient, it's more energy neutral. So that's the first application of bioreactors. So these are bioreactors, only they're bigger, right? They're millions of gallons in volume. Here's another example, energy production. This is an algal photobioreactor, and you're growing algae in this bioreactor. And maybe the question might be, how do you optimize the growth of, let's say it's microalgae, and you're trying to produce lipids that can be used for biofuels. A lot of these microalgae actually have a high lipid content, and you can convert these lipids to biofuels, even aviation fuel. So that is a question that you can ask if you have this kind of bioreactor. You're familiar with bioreactors of all kinds, because this is exactly what we do when we're making wine, we're making beer, we're making fermented foods, right? These are actually bioreactors. You have microorganisms in these containers. And the question might be, how do you optimize conditions for high quality food production? So bioreactors really are everywhere. You can think of the human body as a bioreactor. You can think of your toilet as a bioreactor, your septic tank as a bioreactor. And so we'll define bioreactors more formally, but I just wanted to show that and illustrate that bioreactors are ubiquitous, they're everywhere, and they're also natural, and they're engineered. Here's another example. When you want to produce enzymes and other products, this is a picture from a company called Novozymes. It's a global company that actually use microorganisms to produce enzymes that are useful, like cellulases. So how do you scale this up, and how do you discover new enzymes? A lot of these companies have R&D labs, and they use bioreactors like this, fermenters. So what you're trying to do with bioreactors is really you're trying to examine microbial processes that happen in nature, and you're trying to control that in a smaller system that you can more tightly control. So really, you're trying to get a simulation of the environment into a smaller volume that you can control. So let's put this a little bit in context. And this is one of my favorite graphs that I show to my classes because it really explains a lot of things. So I'll spend some time explaining this graph. This is from Eugene Madsen from Cornell. And it's really how to think about biological experiments. So on the x-axis, let's see if I can, on the x-axis here is the means of producing information. And the means of producing information can be through different experimental approaches. So as you go from left to right, you're going from the bigger systems, field sites, to lab incubations, to pure cultures as you go to the right here. And even in pure cultures, you're going from cells to extracts to genes. So you're going smaller, if you will, right? You're going more into smaller and smaller scales and looking at smaller and smaller components of the whole system. That roughly corresponds to different disciplines too. So ecologists are more in the field, maybe some lab incubations. Biogeochemists are more in the field. But once you get to things like biochemistry, genetics, molecular biology, then you're looking more at these pure cultures or cells or genes, right? So you're here to the right. Now, on the y-axis, let's start with this top one. Relevance to knowledge of microorganisms in field sites. Obviously, if you're in the field site, you have a high relevance, but as you go to test tubes and pure cultures, the relevance to actual organisms in field sites, that goes down, right? Maybe it's not linear, but the point is to show that goes down as you go to the right. What about some of these other characteristics? Well, if you're in a field site, the system complexity is higher. And one of the reasons why you go to a smaller system, like pure cultures, is that you reduce the system complexity. So that may be a positive in terms of controlling the environment and making sure that your experiment is controllable. So of course, your experimental control, how you can interpret what's going on, basically goes up, but also your lab artifacts. So these are things that actually don't happen in nature, but just happen because you're using a smaller system, an artificial system. That also goes up. So you can think of this as biases and errors that happen because you're using a smaller system. So this is one way of thinking about what should I do in terms of my experiment and in terms of my field and the questions I want to answer. So you can go across this whole range. Now, this is value free. It's not saying that being in the field is better than being in the lab. Working with more complex systems is better than pure cultures. That's not the point. The point is to understand that when you make that choice of what system to use, you are making trade-offs and you're deciding in terms of the things that you can control versus the things that might arise because of artifacts. Now, I think it was a few years ago, my students and I actually wrote this book chapter on landfills. And we kind of thought about this too, and we wanted to put that in a very similar graph in the context of landfills. So here on the left are landfills, real solid waste sites, maybe dozens, if not hundreds of acres of landfills. And then on the research side, you might have pure cultures on the right side here. And again, it's the same thing. You might have a low control in actual sites, a higher control when you have these cultures. But this idea of verisimilitude, which means how close is it to reality, again, it's the same thing, right? It's high in the actual site and low in culture studies. But I point this out because we think, and I agree, that the reactors, bioreactors, are in the sweet spot here where you have at least a way of simulating reality, but at the same time having some control. So this is the point of bioreactors, right? So that's just putting this thing in context. So again, what we're trying to do is we're simulating nature, microbial processes that happen in nature in bioreactors. That's really all we're trying to do here. And I know you're doing this also in some of your research and maybe some of your teaching. Maybe you just need to think of it in terms of bioreactors. So the first question is, what biological system do you want to simulate? And it really depends on your field, right, and your area of research. So let's go through some examples. Obviously, we can't cover everything, but some of my favorite examples, and then we'll go through some of the questions that we can ask in these systems. So the first one would be aerobic processes. And my example here is this wastewater treatment plant, and this is called activated sludge. What this is your wastewater goes into this huge tank. It's being aerated. You can see the bubbles, right? It's being mixed. It's being aerated. The microorganisms are inside. And what they're doing is they're taking the organics in the wastewater. It's an aerobic process. So you're taking in oxygen and you're converting the organics, the carbon-based organics, into CO2. And the organisms obviously are doing this so they can grow. So you get more microbial biomass. So that's the first major biological process. It's the most common biological process in the world, probably, because every city that has a wastewater treatment plant would have this system, activated sludge. And how do we simulate that in the lab? Well, these are our lab bioreactors, and you can see that they're not that complicated. You have a plexiglass cylinder. It's plexiglass, so you can see through it. You have ports on the vertical axis here, so you can manipulate where you want to get liquid in and liquid out. It's aerated by aquarium pumps. These are aquarium pumps. You can buy them from the pet store. And then you have maybe pumps that pump water in and pump water out. So it's not really that complicated if you think about it. So I can simulate this basin, which may be 1 million gallons, with this 5, 6, 10-liter bioreactor in the lab. The other stuff in here are timers that control the pumps. If I want the pumps to turn on and off according to a specific schedule. And sometimes you do have mixers too. You can see the modes on top. Okay, so that's one example. Here's another example, anaerobic digesters. And what does that mean, anaerobic digesters? These are systems that are close to the atmosphere. There's no free molecular oxygen. And so you have anaerobic processes happening. In this case, your organics are converted eventually to methane and CO2. And in the middle, you might have something like volatile fatty acids being produced also. Now, how do I simulate, again, these millions of gallons of anaerobic digesters? Well, I can actually just make small bottles like this, make them anaerobic, making sure it's not leaking. And I can capture the biogas being produced and the methane and the CO2 being produced in these gas bags. And then I can measure the gas production, let's say, over time. So again, it's not that complicated. It's really homemade, right? It's DIY. Here's another example, very similar to that first one. This is a real landfill. To simulate this, we would have landfill simulators. Basically, we put the solid waste in the containers. And again, they're anaerobic. I collect the gas in gas bags. And if I had to, I can collect liquid, the juice, if you will, of the landfill that's being produced in these leachate bags. And it was cut off, but there's a leachate bag here. Every reactor has a leachate bag, and you can recycle that leachate. This liquid basically that is produced in the landfill. You can simulate rainfall even by adding more and more liquid into your anaerobic system here. So again, the system is solid waste becomes methane and CO2. So these are not that complicated. They can be complicated by all the controls and sensors that you can put in. But at its very heart, we're simulating essentially what's happening in the big system, like a landfill. Here's an exciting example of a different application. This is a cow rumen. And I think this is, let's see, this is what you use for callos, I believe, tripe, right? And so the question here, this particular experiment that you can see on the left, it's simulating the rumen in this bottle, basically, right? This is from an actual master's student at NC State. I'm sitting on his committee, and his question was, can we change the diet of the cow so that we can actually stop the production of methane? As you may know, cows are one of the major sources of methane globally, right? So it's actually ants and termites and ants, right? Not ants, but termites, because they eat the cellulose of wood, and the bacteria and the archaea in the termites produce the methane. Landfills are another source. Rice paddies are another source. And then cows also produce methane. So in the cow rumen, the cellulose feed become volatile fatty acids. That's what's supposed to be absorbed by the cow, but not all of it is absorbed, and some of it is actually produced into methane. So you can simulate all of that in a small system like this. And again, there are simplifications, of course, but you can ask specific questions. So I like showing that because it's a little bit different. In fact, many years ago, I was invited to the Philippine Carabao Center in Central Luzon, I think Nueva Vizcaya. Yeah, CLSU, next to CLSU. And it was interesting because they were trying to do experiments with the Carabao rumen. Here's another example where you're trying to simulate what's happening in a lake that has a lot of algal growth. This is algae. So this is a photobioreactor. And the diagram here on the right shows how you actually control the light in this photobioreactor. It looks a little bit more complicated. That's because you have sensors for CO2 and sensors for light inside. And also you introduce CO2. You actually bubble in carbon dioxide. So the equation here is light plus carbon dioxide gives you the algal biomass, which you're growing again for things like lipids for biofuels. So different kinds of bioreactors that are possible depending on what you're trying to do. And all of these examples are examples of things that we worked on. Here's an interesting example. This is called a microbial fuel cell. So let's break this down. Actually, some microorganisms, instead of doing an electron transport chain, so maybe they convert an electron donor, a substrate, right? Instead of transporting that internally through processes like glycolysis and PCA cycle, some of them are electroexogens. In other words, they actually give off electrons. So a species called Geobacter does that. And if you put them in a system where there's an anode, they attach that anode and they actually transfer the electrons externally. And if you have a cathode on the other side, let's say a carbon or platinum cathode on the other side, the electrons actually move from the anode to the cathode in this way. So you actually get electron flow. And those electrons then are basically used to convert things like oxygen to H2O. There's a variety of different conversions. So what you have here is actually a battery because you have electrons flowing. And if you put a voltmeter here, you'll see that you're actually getting voltage. You're getting electricity. So from these microorganisms. So you're powering it with waste, right? Organics as your electron donors. And then your bacteria, Geobacter, transfer the electrons, and then you get this circuit going, and you actually can produce electricity. So these are microbial fuel cells. There's a whole generation of these microbial electrolysis cells, and so electrolysis cells. So you actually have MFCs, and you have MECs, and then you have all kinds of electrochemical reactions happening, producing things like hydrogen, or even methanol, or electricity, or other kinds of things. So I just wanted to show an example of a unique system, a unique microbial system. Okay, if we're going to do this, we're going to need to understand some concepts. Okay, maybe before I go into this, I'll show this next slide first. And my point here is that you can actually DIY this. You can do this yourself. It doesn't have to be complicated. Here are examples of something that maybe everybody has done, right? This is a Winogradsky column. You basically put some organic matter, right? You seed it with some sediment. You put it, they say south-facing wall, so it gets the sun. And then you create these microcosms where you have different kinds of bacteria thriving at different levels of the column because of things like oxygen gradients and so on. So for example, here you have iron bacteria on top, and then you have purple sulfur bacteria at the bottom. And then you have, all kinds of things you can add, and you can get different kinds of bacteria. Vinogradski columns are very old, right? But these are bioreactors. So it doesn't have to be complicated. You put in some organic stuff, you put in some sediment, and you're going. Here's an example of a very basic bioreactor. It's basically just a bucket. And then you aerate it, again with, an aquarium aerator. And you have already a bioreaction going in here. Or you can have small anaerobic serum bottles, and these are anaerobic. You can seal them. You probably need a little bit more tools here because you need a crimper, you need a cap, and so on. But once you get those, you can get some experiments going. So the point of this is that you can DIY this. Now we have to go through some concepts to understand how you actually do this. So there are, in general, 3 ideal bioreactor types. Okay, the first is called the batch reactor. So the batch reactor is your bucket. You basically put a whole batch of liquid, and then you let it react. It's like making a smoothie, right? If you have a blender, you put in your ice, you put in your water, you put in your, banana, and then you blend it. And that's the reaction. So it's a batch because you put everything together, and then you let it react. That's a batch reactor. And then when it's done, you pour it out. So it's batch in, batch out. If you look at the concentration over time in your batch reactor, so here's time on the x-axis, and this is concentration of whatever it is that you're trying to look at. And this is let's say this is organic matter, and you have microorganisms decreasing the concentration of the organic matter in your reactor, then they would decrease over time like this. The exact shape obviously can change depending on the kinetics of the equation, of the reaction, but it's going to decrease over time like this. So when now you don't have any more organic matter, you can now throw out the whole thing, right? Okay, that's the batch reactor. The next type is the continuous reactor, sometimes called the continuously mixed reactor, sometimes called the CSTR, continuously stirred tank reactor, where you have a pump or continuous flow of liquid coming in and liquid coming out. And so the water level stays the same because it's going in, it's going out at the same time. And this is continuous. This is a little bit different because if you look at the concentration, let's say, of organic matter, it's always low inside this system. And it's completely mixed. So at any point in this reactor, you can sample here, 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 and it's the same concentration as also what's going out, right? So that's a CSTR. Then the third type is called a plug flow reactor. And you can imagine this as a long tube. And essentially, a plug of liquid travels through the tube and goes from left to right. And as it travels, you get the change in the liquid. So for example, your organic matter goes down over time or over length. So in this case, your x-axis is length, and here's how your concentration goes down. Now, mathematically, the batch reactor and the plug flow reactor are very analogous. And you can see it from the curve, right? They look the same. But these are some of the ways that you can think about how to set up your system. Okay. And like I said, here are examples of batch reactors, right? You put things in, you let it react, and then you see what happens as that reaction is going, and then you can take them out and start over again. Okay, so there are some basic concepts if you want to talk about bioreactors. So the first is what we call the hydraulic retention time, or HRT. So here's a continuous flow reactor. with a certain volume V, and it's going in at a flow rate Q. That's just our notation for flow rate, right? So it's Q coming in and Q coming out, and those two flows are the same. Now, let's say cubic meter per second or liters per hour, so that the liquid stays constant, right? Okay, so HRT is the amount of time that the liquid stays in the reactor. And it's given by this equation. So tau is HRT, volume divided by flow. So how big is your reactor, your actual volume, divided by the flow gives you the time. So we can put in the units, for example, cubic meters divided by cubic meters per hour. Cubic meters cancels out, the hour goes to the top, and so tau is in hours. It's the amount of time that you have the liquid inside the system. So in the batch reactor, it's how much time basically you let it stay there, right? You're just putting it in there and it's cooking. That's the amount of time. But in a continuous flow reactor, this is the equation. So this is important because that determines how much you can put in depending on the size of your reactor. So in most engineering, you want this to be low, this number, HRT, because that means for the same volume, you can have a lot of flow, right? You're treating a lot of water. That's what it's saying. But this amount of time should at least be as large as the amount of time that it would take the microorganisms to do their reactions. And typically that's fast enough to be in the order of hours, okay? But not minutes, maybe not seconds, obviously. Like, for example, you cannot have flow going in and then going out without enough time for the microorganisms to react. So that's the first concept. Usually here's where I stop and, I ask my class, okay, is that clear? Okay, but we'll have a quiz later on. Okay, so make sure you're taking notes. Okay, so the next one is called the SRT, or the solids retention time. Now, this is a little bit more complicated, but let me simplify it. What this is the amount of time that the solids, or the biomass, microbial biomass, stays inside the system, right? So it's the amount of time that they're allowed to be there. And the reason this is different from the HRT is because you have a solid separation step. In other words, in this reactor, for example, I'm mixing this, okay? So the microorganisms in the liquid goes out, but I have a sedimentation tank, and here's how I separate my microbial biomass. It settles to the bottom, and I can pump it back and recycle it. And then the liquid goes out. Because of this separation step, the liquid flow determines the retention time of the liquid, but the solids can stay there for a long time because they actually can be recycled, right? Okay. Now, to simplify this, you can just think of this as the amount of time, the amount of biomass, okay? Not concentration, but total amount. Let's say in kilograms or milligrams. The amount of biomass you have, the inventory inside the reactor, divided by the amount that you waste per day. That gives you the amount of time that the biomass stays inside. Let's do maybe a simpler analogy. The population of UPLB is, I don't know, probably, I'm going to guess 20,000, okay, students, undergrad students. Okay. So you have 20,000 students at UPLB. And every year, 4,000 students go in. And every year, 4,000 students graduate. Okay? 20,000 in the whole university, 4,000 in the freshman class, and then 4,000 in the graduating class. That way you keep that 20,000 always inside the system, right? On average, how much time does each student stay inside UPLB? How much time does it take for a student to graduate, given my numbers, right? And the answer is, inventory is 20,000. And then the wastage, the amount of solids or the amount of students leaving is 4,000 a year. So 20,000 divided by 4,000 a year, 5 years. On average, each student stays at UPLDE for 5 years. That's what this is saying. Now, why am I harping on SRT so much? The reason is that SRT is related to growth rate of the microorganisms. And so before I go to do that, I think, this concept of growth rate is basically, right, the amount of how fast organisms grow. So we call that mu. One of the more common kinetics that we use is monokinetics. And so this is what it looks like, right? This is substrate on the x-axis and mu on the y-axis. And this is how it looks like. As you increase the substrate, the food, the growth rate, organisms actually grow first slowly, but then they keep increasing. But then they start to slow down until they reach a maximum. In other words, it doesn't matter how much food you give them at this point, they can only grow so fast, right? It's like, when there's little food, you're here, but when there's a lot of food, you're here, okay? And that is the Monod equation, okay? So I'm not going to quiz you on this equation, but I just wanted to show you one representation of mu. Now, SRT is related to the growth rate. I'm now relating this to a bioreactor, and this is given by this equation. So we won't derive this, but the main thing here is that the growth rate mu is equal to the inverse of the SRT, 1 over theta C. Theta C is SRT, plus B. B is the decay rate or the death rate. So let's say the death rate is 0, okay? Just so we can simplify this equation. What this means is that if I have a long SRT, like 1 over a large number is a small number, right? Which means if I have a long SRT, the growth rate can be small. Now let's think about that. If you give me a lot of time to stay inside the reactor, then the growth rate can be small. You will survive inside this reactor because the retention time is high. Conversely, if the retention time is low, let's say 1 day, then this growth rate is 1 per day. In other words, you have to grow that fast. You have to double in 1 day, because if you double in 2 days, you're going to be washed out, right? You're going to leave the reactor. So in other words, the SRT determines who grows inside the system by being a threshold, if you will, right? So organisms need to grow as fast as the SRT allows, okay? So I'm basically going through these 2 basic concepts of HRT and SRT because I wanted to give a little bit more framework, right, on bioreactors. Okay, so questions you can ask. What are the types of research questions that you can ask? And I'll give you, again, 3 examples: aerobic, anaerobic, and energy processes. Okay, so one question is looking at the interaction and competition between 2 kinds of organisms in activated sludge. The first kind is, and this is again wastewater treatment, the first kind are flock formers, they form flocks. And the second kind are filaments or filamentous bacteria. You can see some examples here, right? And the problem is when this competition favors one over the other, especially the filaments, you get all kinds of problems, including this problem called bulking. In other words, the biomass doesn't want to settle in my clarifier or my settling tank. Okay, so that's the problem. And how do I study this problem in the bioreactor? Well, I can set up a bioreactor experiment. You can see how this is set up. That's my wastewater feed. This is my bioreactor. And because I know that my filaments and flock formers have different monokinetics. So the filaments have the pink line here. The flock formers have the blue line. So depending on my substrate concentration, if I'm in this region right here to the right, then the flock formers are going to win. They're going to have a higher mu, right? If I am here to the left, the filaments are actually going to win. So what this says is that if the substrate concentrations are low, the filaments are going to win. If the substrate concentrations are high, the flock formers are going to win. Now I can change the substrate concentration by changing the feed or by changing the bioreactor type. And so here's an experiment where I changed the bioreactor type. And lo and behold, I was able to say yeah, if I do it one way, which is a CSTR, continuous flow, I'm going to get filaments. And if I do it with a batch reactor or a plug flow reactor, I'm not going to get the filaments. So here are examples of, the same, everything's the same, SRT, HRT, feed, and so on, inoculum. It's just that the way I operate the reactors is different, and that influences who grows in the bioreactor. So I can do things like, ecology, right, with bioreactors. That's one example. Another example might be nitrogen transformation. So here is where we are now in terms of nitrogen transformations. So obviously you have ammonia oxidation, ammonia to nitrite, nitrite to nitrate, that's nitrite oxidation. There's a few new pathways here that maybe are not quite in textbooks yet. One is called COMAMOX, which is complete ammonium oxidation. So it doesn't stop through nitrite. It goes all the way from ammonia to nitrate. The other one is ANAMOX, anaerobic ammonium oxidation, where a group of organisms take ammonia and nitrite and convert that to nitrogen gas. So that's also quite new. And then you have this denitritation and obviously the classic denitrification pathways. And so the question is, how do we start understanding some of these processes, right? So here's an example where you run bioreactors, and what you do is you turn the air on and off. So you have different aeration to non-aeration time ratios. So like 1 hour on, 1 hour off. 1 hour air on, 3 hours off. So you're cycling through aerobic and anoxic periods in the reactor. And then when you do that, you start looking at the dominance of specific ammonia oxidizers and nitrite oxidizers. And so here's the summary. Essentially here, what this is saying is it doesn't matter what the aeration ratio is, right? It's dominated by Nitrosomonas and Nitrosococcus mobilis. And then they're about the same, right? But if you have different aeration ratios, you actually have an inhibition of the NOB, the nitrite oxidizing bacteria, in this case, Nitrospira, the dominant NOB. So by changing conditions in your reactor and comparing them to each other, you can actually say something about what's going on and the impact of environmental conditions on your system. Okay, let's go to an anaerobic example. This one is looking at food waste. What we want to do here is we want to take extra food waste and co-digest that, convert that to energy like methane. And here's the pathway. Probably many of you are familiar with this, right? You take your macromolecules and you ferment them to fatty acids and alcohols and to short-chain fatty acids like propionate and butyrate. And then they're further oxidized to acetate, right? Amino acids to acetate, sugars to acetate, fatty acids to acetate, and also H2 and CO2. And then finally, you have the methanogens that take either H2 and CO2 or acetate, and they make methane. Okay, this is what's happening in the cow rumen that we mentioned earlier. This is what's happening in those anaerobic digesters. So the idea is we want to understand how to optimize methane production, because this methane we can capture and we can use as energy, right? We can use this as fuel. So we can set up reactors, and these different reactors have different types of substrates and different microbiomes. We manipulated the microbiomes, and then we asked the question, who is doing what based on which pathways are being turned on in this network. And here's the overall results for that experiment. Essentially, these are showing, the different colors are the different types of substrates. So this is waste activated sludge. The blue one is carbohydrate. And then the red ones are lipids or fat. And you can see how the different microorganisms are connected to each other and how they're also different in that all the lipid degraders are here, all of the waste activated sludge degraders are here, and so on. So you can actually ask who's doing what by doing these kinds of experiments. Okay, some other comments. if you're going to do this, we have to think about sampling because you want to measure, right? You just don't want to set them up and then leave them. You actually want to measure something. So here's one thing. Depending on the need for monitoring and the sampling size, you might be forced to have bigger reactors. This is about the biggest, reactors we have in the lab. These are drum reactors, almost 55 gallons. And so you can actually, take a lot of sample if you need to. And then our smallest reactors are these serum bottles, 150 mils. Okay, so sampling is a big issue. And then the measurements, and that depends a lot on what you're trying to do. If you're trying to measure organic matter, then you need to measure BOD and COD and understand how much sample do you need to measure these, right? So if COD, maybe you need 2 mils. If you take out 2 mils every day, if you only have 10 mils of a reactor, then you're going to run out of volume, right? So you got to think about some of these things when you set up the experiment. So we won't go through all of these, but basically these are all the different things that you can measure. We have a whole research effort in particle size distribution using all kinds of techniques. And then obviously looking at the microbial aspects with microscopy and molecular methods like DNA and RNA. So what I wanted to show was basically that, one, I hope I convinced you that bioreactors are one way of investigating, a powerful way of investigating a variety of different processes that you might be interested in, and they can be engineered or natural processes. And hopefully you're thinking about using them in your research or using them in your teaching, wherever you are. And hopefully we have time, we can talk about some systems that you're interested in. So multi-step approach experiments, I think you want to choose the process that you want to study, and you want to define the questions that you want to answer. And then that will determine the design of the bioreactor and the experiment. So the questions asked define the tools that are needed. The tools including the bioreactor itself, your measurement tools, your microbial analysis tools, whether you need this type of molecular method or this type of molecular method. And then finally, when you hear bioreactors, you might think, oh, these are complicated systems and so on. But I hope that I've shown you that you can take a bucket and you can make that into a bioreactor, right? It's a matter of simulating what you want to simulate. And so this is really hands-on, and you can see a lot of the pictures are actually things that we just make in the lab. It's DIY, it's do it yourself, and it's a lot of fun. So hopefully it's an introduction to bioreactors, and then hopefully we have time for questions. Thank you very much.